So I request Dr. Rakesh Mishra to kindly.
Thanks, Chandak, for the introduction, and I really thank IAMD for this event. I have heard about this event from Chandak before, so I'm really thankful for letting me in this community, which is very, very important for what we do at the IGS. And I'm going to talk to you about how we are exploring how the cutting edge science can reach to patients at a very early stage, as early as possible in Indian context. So I will spend 2 minutes in telling you what TIGS, which is Tata Institute for Genetics and Society, is. So it's a unique initiative of the Tata Trust to support application of science and technology to solve societal problems. We are a non-profit organization funded by the Tata Trust. And our approach is to use cutting-edge science and technology to solve societal problems, as I stated. All our programs are focused on direct societal relevance and a program-driven organization which stakeholders are on board from the start. So we like to work with people like you because rare genetic disease is our one of our interest. Uh, yeah. So the vision of the institute is how lab activities reach society. So we define the problem and uh, do the gap analysis, prepare the project, strategizes, and then we develop the solution through collaboration, through our R&D, and then evaluate the system, and then take it for implementation through a tech transfer or engaging stakeholders. If there's a policy gap, we try to address that. And our whole aim is that the solution has to be economically viable and socially acceptable. If it is not the case, then we bring acceptability and affordability for larger section of society by various other means that are possible. So to do this, we have uh, uh, taken a strategy where collaboration is our very, very crucial strategy, where we collaborate with NGOs, supports groups, trust, with industry partners, with research institutions to complement what we don't have as a facility or expertise, and the clinicians and hospitals and government agencies, because without working together, our solution will not be reaching where we want it to reach. And we are located in the Bangalore Life Science Cluster. We have the same campus as in STEM, C-Camp and NCBS, which is in a broad campus of University of Agricultural Sciences in North Bangalore. And this institute has three programs, infectious diseases, crop improvement, and rare genetic disorder. And I'm going to talk to you about what our initiatives are in RGD. So here we, one kind of activity is to take a stock of the situation, to make database, put information together, bring stakeholders together, and set up outreach programs. And then other initiative is to use molecular and cellular technology for developing diagnostics, which can be probably be uh, used for even screening and develop various models, and then of course work for the affordable therapies, including mRNA therapy, which and CRISPR-based in vivo gene therapy and drug repurposing and so on. I'm going to very briefly tell you, just to introduce what we are doing. The purpose really of my presentation here is to let you know what we are doing, also let you know that we are with you and we need your help and involvement in many of the activities we are doing. As you will know, it's very clear, it will become very clear. We are not a typical research institutions who does the research, quality research, publishes and moves on. Publication is our collateral. We want to solve problem to reach people. If it's not reaching, then we consider it as a failure. So with that background, so this is one of the activities that we are doing, where we have set up a database for Indian genetic RGD database, where we try to collect all the information that is available of various disease, their genetic links, and many other things that are known about them. We call the genetics. And here is a link for this. You'll not be able to connect with it today, but I think in a week or two, it will be available, and we'll share with you the link, and we'll need your input on this, because our ambitious goal of this database is to bring in collaboration with the clinicians and incorporate in safe way clinical data and give the free and secure access. It should be interactive, and we want to incorporate tools which will be used for diagnosis and counseling. So it will have some utility beyond what is required for the scientists to do research in this domain. Our other initiative, as I mentioned, was the diagnostic, and we are actually setting DNA-based diagnostic platform using all the DNA nucleic acid technologies to do diagnosis in a relatively disease agnostic manner, whether it's infectious disease or rare genetic disease and so on. But some of the special efforts that we are putting, one of them is developing digital PCR technology for the SMA. And just very briefly, we have the multiplexing of 5 sets of primers, which covers the key mutated regions. We have established this assay, and now we are moving forward for the validation with clinical samples that we'll be getting from CDFD and CCMB. And once that is there, I think in next few months, this will be handed over for the implementation with whatever regulations that are required. So that is one kind of thing we are doing. We are also using non-DNA technologies here. This is proteomics-based technology. So actually, this is, we are focusing for diseases where lumbar puncture is the current method of doing, which is very complicated, and therefore it doesn't reach people that efficiently. So these are rare diseases, 13 primary rare disease group, which are called monoamine neurotransmitter defects. They are metabolic disorder, and the rationale is that this is a pathway, and if the enzyme is defective, then this part of the metabolites will be accumulated, they will increase, and downstream will be reduced. And so if we can pick these metabolites, so we have picked some 30 of them, and with their standards, we have now established that we can actually measure in the using the LC-MS technique. And our aim is to use dried blood spot, bring it to the lab, and then in matter of a few, maybe 30, 40 minutes, you can do this analysis. And we are collaborating for this Indira Gandhi Institute of Child Health for the samples, and for other diagnostic centers. And again, once the patient samples starts coming, I think in few months, we will be ready to start a regulatory process to implement this. So in addition to that, being in campus and taking these privileges that are around, we are setting up a patient sample biobank with the NCBS, and also making iPSCs, and I will show you a slide on this of patient cell lines, and also making mouse models, which will be freely available to the entire community in the country to use them for developing, understanding disease and developing drugs or other interventions. So this is what I was saying, we have established the stem cell technology, whether it's a patient sample, healthy individual or patient. We take the sample, convert into iPSCs. If it's required, we do a genome editing technology, and then they're ready for screening as a disease and dish model. We have not quite reached, but we are also developing various organoids, and whatever lead we get, they can be used as for preclinical purposes in the mouse. What I want to say is that we have established this platform, and we have put the timeline that when the sample comes, in 2 months we reach this point, in 3 months we reach edited points, and in 6 months we are set for using the cells for the purpose which they have been initiated. So so this is already in place. So I will give you some examples. So anytime any researcher wants to make a iPSC, in few months' time we can make them available. So this, for example, this is in CMC Vellore collaboration. So this is a very rare genetic disease called brittle bone disease, osteoporosis imperfecta, for which we have made we have made iPSCs, and you can make the you can see the results that they are reliable iPSCs which can now be tested. This is again a sample for the Pompe disease, which we take the cells from the from the patient and convert, establish the iPSCs, see the mutants, and you can see they are, we do all kinds of control, and now these cells are available for testing anything which can be used as a drug or something. So that is about the setting up the cell-based technologies, and that is in place. The other very ambitious technology, which is we have made significant process in, that is the mRNA-based platform, which is really the basic thing is such that we can then use it for biotherapeutics, in vivo gene therapy, and vaccine, and so on. And we have made modules of this. Stage one is mRNA design, gene synthesis, mRNA generation, lipid nanoparticles, and so on. Then stage two is validating what you make. The stage third is the testing for the toxicity, and then of course GMP facility, and then goes for the clinical trial. And here we have established all these steps. So, and we have set the timelines when the day we have the sequence that this is the gene that we have to work on. In 12 months, it will be ready to go for the GMP facility manufacturing and clinical trial. And we have partners who can take this step once we reach there. So the initial aim of this is to use mRNA therapeutics for the candidates lysosomal storage disorder like Pompe, Fabry, Usher, and gene myopathy. Idea is to use mRNA-based therapeutic intervention for monogenic rare genetic disorders in India. And with this, we should be able to replace the costly enzyme replacement therapy with an affordable mRNA therapy. What we are talking about is a few hundred rupees per dose. So you can imagine the transition that will happen, the transformation that will happen, that it will be available with no hassle of injections and so on. So even hospitalization might not be necessary for them. So idea is that because this is a functional therapeutic protein made inside the body, it should be much more effective than making them ex vivo and administering to the patients. And we want to project this as a really a disease agnostic, affordable alternative. The day you know this is the gene you want to fix, the rest everything is automatic once the template for the transcription is decided. And our larger goal is that once it's fully operational, mRNA-based in vivo gene therapy is one of our target that we are preparing the strategy. In vivo gene therapy is very different from the current regular gene therapy, where we put in the lipid nanoparticle, the template, and also the CRISPR and guide RNA, so that it can go and inside the human, inside the patient body, do the genetic corrections, and which probably will lead to immediate relief. As we see, this is so simple that once in place, even if it is benefits maybe 10%, 50%, whatever it can be, it will be very, very useful for the populations like us, where we cannot imagine 20 crore and 30 crore treatment for one patient. And then we also look at some point this mRNA-based monoclonal antibodies for cancer or for the vaccine for the animals or plants, because this is going to be really affordable technology. Few hundred per shot means everybody can use it, and it can be used for large scale problems which our country faces.
Wind up in another minute or so.
Okay, yeah, so we'll do that. This is just basically telling how the technology works. So you have a mRNA, you can package and put in the animal or cell and test, and this is to show that we are already in our lipid nanoparticles are expressing, and this is to show that this is a GLA, which is a Fabry disease gene. You can see that when we produce this, there is expression for up to 120 hours, I mean several days. And this is a GLA mRNA that you can see. And here, this is another test for say, GNE and for the GA. So which means we are able to, whether it's Pompeii or Fabry or Gini, we are able to now make this RNA, make this particle which are now expressing in the cells, so they can go for the toxicity testing. These are other our efforts which I'm not going to describe. There are several other things we are actually initiating because once technology in place, we can do many of those. And the last part is demystifying the rare genetic diseases. It is very important because there is a social angle involved to it. So we are making information, working on policy, making multi-stakeholder engagement, and doing this in a really very active manner. This is our annual program. Next time we'll share with you this, and we'll expect most of you to be there, where we call it redress, where we involve lots of stakeholders, patient groups, clinicians, hospital people, researchers, policymakers, to be in one place. And so we're making various postcards, various poster series, and some of them we sometimes do deep science, sometimes more popular, and try to take this message to as far and as many people as possible. Purpose, as I said, is that we look for people to join hands to make a difference. We have a large number of partners, and we want this place to be filled with organizations like yours and many of you who are interested in any kind in rare genetic disorder, because this is something where it is a very unique problem. It requires new strategies and new approaches, and all of together, when we join hands, we can make a difference, because science is now there. So we can now make that science reach people. So that's the aim of my talk. So thank you again for giving me chance and listening to me. And thank you, Chandak, for tolerating me crossing the time. I'll now stop sharing, and if there is anything to discuss, I'll be happy